Hello. See that people are connected. Sorry, all of a sudden when even though I ran a Zoom session earlier today on the same computer, all of a sudden it was saying it needed to install updates. So I shouldn't have waited to the last minute to try to start my Zoom session. All right. So, let me just see. I was going to share my screen, but I got to make sure I was done and ready to share it. Let's see. Okay. So I'm going to share my screen here and I'm going to show you, and then we can sort of go through things. So I basically took the I took the exam and then I took away like all of the information from the questions, but letting you know what the topic headings are. Okay. So there's two main questions that are like the short answer or like the one to 2 word answer questions. We really didn't learn about enzyme names too specifically. So it's not like you're going to see those detailed things where it's like step number, enzyme name, substrate, product, whatever. It's literally just more of the, okay, I'm asking you a question, it's a fill in the blank, it's answering with a 1-word question, things like that. So there's a laundry list of ones that are about electron transport and oxidative phosphorylation in mitochondria. And then there's a laundry list about electron transport and photophosphorylation in chloroplasts. Okay. So that is about half of your exam. So, think about questions like, who is the final electron acceptor in mitochondria? What is the rate controlling, what is the enzyme responsible for carbon fixation in the Calvin cycle? Thinking about 1-word answers for those things. So about half of your exam is that. Then there are 5 short answer questions, and you probably can sort of figure out what these are. There's 2 questions on thinking about equations for mitochondrial biochemistry and chloroplast biochemistry. So again, you had some homework questions that asked you to have a main equation that maybe like showed you how things connected, right? And then there were homework questions that dealt with how electrons flow and how that's different in mitochondria versus chloroplasts. And then sort of a question thinking about, these are a lot of compare and contrast type things, but thinking about chloroplast versus mitochondrial anatomy. Okay, so those are kind of the 5 short answer questions. And remember last time there was a laundry list of questions that were sort of new questions, think outside the box. This time there's 3 of them that are worth 5 points each, so a total of 15 points for those. So that's sort of the, I wrote the, I had the exam and I just deleted the questions so you could see the topic headings. So you sort of know what things are. So I'll pause for a second, see if you want to ask any specific questions on this, and then we can sort of go through and look at the review sheet and talk about things from that. No? Okay. So let me go over to Blackboard here. And let's go first, review session or study guide. Right. And so the study, I just pulled this off of Blackboard. So the same thing that you guys have. So it kind of walks through these specific things, shows you homework questions or things like that that are relevant. So I thought we could just sort of talk about that. If there's anything you'd like me to discuss in a little bit further detail, I figured we'd start first with Unit 5. I have up available here sort of the homework key, as well as sort of lecture slides. So, as a hint, this is sort of the equation slide, right? So thinking about the main equation and how we sort of connect pieces to that. Let me come over here. Equations. 2 main equations to describe fuel oxidation and carbon fixation. So I guess I should say the way that this learning or this study guide is set up, it's really a compare and contrast one. So it's sort of, we'll highlight if there's things that ask you to compare and contrast mitochondria and chloroplast. So that's sort of these equation pieces. So, and maybe we can do that. Let me go over to, and I guess it's really on here. We do both of them. So this is really the compare and contrast. So it shows the mitochondrial fuel oxidation and then the chloroplast fuel production. So that's kind of what that is. So I would know these equations, how things are connected and sort of do their biochemistry. Let me see what else I have here. So fuel oxidation and then carbon fixation. So both light and dark reactions in the chloroplast. There's only a fundamentals quiz on electron transport and oxidative phosphorylation. And I've tried to start sort of getting away from calling it the dark reactions. So I'm going to start trying to do it here. Then I'm not going to use the word dark reactions. I'll say Calvin cycle, because I think it can get confusing because all of a sudden you think that these are things that happen when the sun is not out. And hopefully I've convinced you that is not the case. The Calvin cycle is not something that happens when it's dark out. The Calvin cycle can only happen when it's light out. So again, sources of electrons are from acetyl-CoA as a branch point metabolite, and that was stuff that was in notes. I'm going to pause there for a second, see if anybody has any questions or something you want me to go a little bit more deep on. So let's see, I'm going to just kind of flip through homework here. Right, here's our equation. So again, that's, I would know that. I would know that equation. Fundamentals pieces for ETOP. These may appear as sort of individual sort of questions when we're thinking about electron flow, right? Pathway for electrons. I would definitely know pathway for electrons in electron transport in both mitochondria and chloroplast. So you have sort of pieces like this. So diagramming mitochondria. Let me go over to sort of notes here. And again, when we did this stuff with chloroplasts and photosynthesis, it was a good opportunity to sort of review as we did this compare and contrast. So this is kind of an important figure to make sure that you know, being able to diagram the different things that we see with mitochondria versus chloroplast, all the different names and so forth. Let's see. So again, that's that organelle structure, compare and contrast. Go get the kitty. Well, I don't know what the kitty's doing. So again, we've got these transport issues. So there was a homework question that kind of, and I don't know why my homework wasn't finished here, but I know it's finished in the notes, thinking about the different things that we needed to transport. So I would, more than this homework piece, I would go by sort of what I have here, making sure that the language that I have here, in case there's any, if there's ever any discrepancy with what is in sort of a homework key that I post and my notes, go by the notes, okay? Unless you feel like it's wrong, but there may be things that I just clarify a little bit, because I made that homework key probably 2 or 3 years ago, and I probably need to go through and make sure to correct things that have been clarified. I shouldn't say there's probably not anything that's wrong per se. Is it just I may have clarified it? Electron transport. Again, semi-detailed questions about electron transport in both mitochondria and chloroplasts. You know, be able to think about the order of things. You know, who are electron carriers? In both of these learning units, we kind of did some walkthrough slides. So being able to think about things like this in terms of, you know, where electrons come from, who they go to, whether or not it's a complex that translocates protons, things like that. talked about light harvesting, in our notes. So kind of coming over here, how light is harvested. I don't know. Let's see. Let me go back here. Generation of specific products. So you may see, and I know you saw this on your quiz today, sort of a lot of compare and contrast. So this species in mitochondria as to this species in chloroplast. So being able to sort of, hang on, a cat is getting into something. Get down. You're being bad. Okay, Charlie, get. It's okay. I have the kitty. It's all right. We're going to show everybody. This is Leo. And Leo is a bad little kitty. Leo gets into stuff. All right, Charlotte, ready? Go get your kitty. Get your kitty. All right. Let's see. ATP and carbohydrate synthesis. So steps of oxidative or photophosphorylation. So being able to think about how ATP synthase works. Some questions might, have to do with the details of, that loose, tight, open sort of we call that a binding change mechanism. So that's the ATP synthase structure and binding change mechanism. Comparing and contrasting the pentose phosphate pathway versus the Calvin cycle. There's homology that we have here. Rate determining steps. Thinking about the compare and contrast there. Little bit about, we talked about carbohydrate synthesis routes. We didn't do a ton in lecture, but I do want you to remember things about synthesis of carbohydrates, where, and this is sort of reviewing things that are important when we think about amylose, right? That is generating an alpha-1,4 glycosidic bond. You do not need to remember, names of enzymes or things like that here, or that, ADP glucose is specific for amylose synthesis. But think about the different things that are synthesized in plants. We've got starch, right? That is going to be a storage piece. We've got sucrose, which is a transportable fuel, and then cellulose with that beta-1,4 linkage is going to be an important building block. Let me see here. Pathway regulation. We really didn't talk too much about regulation in electron transport and oxidative phosphorylation, other than talking about regulating ATP synthase by a pH-dependent inhibitor. So if you didn't remember where that piece was, let me find that here. So this is all that we talked about. So this is a pH-dependent inhibitor. And all of these things should sort of make sense, right? If we are doing a lot of electron transport, what we're going to be seeing is an increase in pH in either the matrix or the stroma, right? And so we can have that sort of affecting things. So in this case, if we've got, this inhibitor domain, it's going to be, we don't want to be inhibiting ATP synthase if we're actively doing electron transport. But if we're not doing electron transport, we want this inhibitor to be in sort of this dimeric state so that we can be inactivating ATP synthase. Let's see. Thinking about how photosynthesis is regulated in the day versus the night. That's not something we think about with fuel oxidation. Talking about Rubisco regulation. So there were several videos that got put out in the last few days here, thinking about how we regulate the Calvin cycle, right? And so that's FBPase, SBPase, and then thinking about PFK regulation. And then this aspect of regulating photosynthesis and CO2 acquisition in time versus space. So this idea that there's certain plants cells that are going to sequester CO2 and store it as malate. And the reason that it does that is so that we can avoid photo respiration. So that we don't want to have to use oxygen with Rubisco. So again, that concept of photorespiration is there. Pathway integration. So again, this is thinking about, whether it's a phosphorylation cascade or a redox cascade. We really didn't talk about this, so don't worry about this. We didn't talk about roles for mitochondria this year, so you won't see any questions on that. And we really didn't talk about mitochondria and disease. So you can sort of, actually this pathway application stuff, we didn't talk about obligate aerobes. Remember, there was 2 questions I told you even on your homework, don't worry about. We didn't get a chance to get to it. Interesting stuff, but you're not going to see any questions on your exam about that. So I just kind of like talked through and read you the study guide, but are there aspects of different things that you want me to go through? What do you think would be helpful for me to do with you guys right now? Do you want me to flip through slides? Do you want me to look at the homework? What do you want me to do? Lauren?
Can you go through the quiz today?
Yes.
The 2 questions about the homologs or whatever tripped me up about the mitochondria and the
Okay. So let me think the best way to do this. So what I'm going to do is I'm going to, I think, open.
If you want to open mine, you can.
That's okay. These should be the questions that I had. So everybody had the same 10 questions. You may have had them in a different order. You may have had them with the answers sort of mixed up. That's how Blackboard does it. So, and hopefully these are the questions that you had. I actually didn't look through. Do these look like they're all the questions that you had? So I'm trying to think if there was something about a home. I don't remember there being a question that said homolog.
It was the there's one at the beginning, the O2, like the
Okay.
I guess.
Got it. Yep. So why don't you tell me? So here's the right answers for these. Hopefully you guys can see the answers on your own Blackboard quiz. But so tell me which one you want me to go over, Lauren, and I'm happy to do that one particularly.
51 and 52.
Okay. So O2 is to mitochondria. So what is O2? And I'm going to ask you, I'm going to put you on the spot just because you asked this. Okay. What is oxygen to mitochondria? It is the
Final receiver.
final electron acceptor, right? So what then is the final electron acceptor in chloroplasts? You can see the answer there.
NADP is the final.
NADP plus. Yep. So that all of these, this is how when you make, Blackboard's kind of a pain in the ass. I have to create it in Word. I have to have it be a text file where I label the correct answer with an asterisk before this. I have to import it into another program that creates a test pool, and then I need to import it into Blackboard. It's not fun. But does that make sense now?
Yeah.
So was the wording just confusing, or?
Well, I was more thinking that mitochondria can run on oxygen, whereas plants, I don't know. My mind went to CO2 for some reason.
Got it. Okay.
I wasn't thinking what O2 is necessarily to mitochondria.
Got it. So you could have been thinking mitochondria is a reactant in mitochondria. So you were thinking what is a reactant or something that's on the left side of an equation. And that's not unfair, but then there was several things that could fit in that pot. So we have to then drill a little bit more deep in that and say, okay, specifically, what is oxygen in mitochondria? Oh, it's the final electron acceptor.
Okay.
Any other, since I have this up, is there any other questions? I'll kind of scroll through slowly if there's things that people wanted me to address in terms of if you didn't understand the answer.
Will you do 52? So
yeah, oh yeah, you asked me 52. So H2O is to chloroplasts. So what do you think about water and chloroplasts? How is water used in chloroplasts?
Reactant. I don't 100%.
So let me go over here. I'm going to go over here for a second, and I'm just going to pull up a very specific slide. Let's look at it here. So where do you see water here?
I know it helps start the like gradient. It gets broken apart.
So anyone want to chime in? H2O is specifically what?
Electron donor.
It's the source of electrons for the electron transport chain. Okay, so if I come back over here to my questions, Lauren. So what is the source of electrons in mitochondria?
There it is. Na.
NADH. Now you notice I didn't have FADH2 because that could have technically been a correct answer as well. So I didn't have that specifically on here, but that is kind of the source of electrons.
That makes sense. Thank you.
I wanted a second with Lauren. I got those two questions wrong too, because I was thinking the same thing. Like it sounded like we were comparing them as reactants in the equations.
I did the
So,
same.
and you're right, they are both reactants, but then there's multiple things that are reactants. So because there's got to be one answer that's correct, it's like, okay, specifically, how is O2 a reactant in mitochondria and how is H2O a reactant in chloroplasts? Yep. But maybe if you saw one reactant that was there and you're like, oh, that's got to be the answer because I see it as a reactant, then you sort of like, hey, answered that question, moved on. All right, here's 3 more. Let me know if anybody wants me to go over any of those if you got that one wrong.
Or 7. I couldn't remember. During night, is PFK inhibited or is it activated?
It's going to be activated. So let me go to that slide here so we can sort of look at that. I know exactly what slide I'm thinking of, right? So there's this guy. So when we're thinking about what happens during the day, okay, we're thinking about light reactions versus dark reactions. Actually, well, I guess I thought I had a different slide here. Sorry, I'm flipping quickly because there was one that I thought was different, but I guess not. Okay. So when we're thinking about the light reactions again are going to be active just by virtue of the fact that there's light. If there's light, then the light reactions, electron transport in chloroplasts is happening. Dark reactions are going to be active because we are going to have a high pH in the stroma, right? Because we're moving protons out. That is going to allow Rubisco to be active because we're moving protons out, we're moving magnesium in. So we're going to have high pH, high magnesium in the stroma when electron transport is happening in chloroplasts. And because electron transport is happening in chloroplasts, we're also going to have SBPase and FBPase be active. So all of these things, light reactions and dark reactions, are active at night, or I'm sorry, active during the day. And because of that, we don't need to be breaking down carbohydrate stores. So we're going to be inhibiting glycolysis. PFK is the rate determining enzyme of glycolysis. So we're going to be inhibiting that. Opposite is true now at night. Now it's easy to say that the electron transport chain is going to be off or inhibited because there's just no photons, there's no light. So, but once we do that and there's no electron transport, then the pH is going to drop in the stroma because there's going to be lots of protons that are still in there because we're not moving protons out. We're not going to be moving magnesium in. Both of these are going to help Rubisco be quiet. And then plants will also synthesize this transition state analog at night. So 3 things are going to ensure that the dark reactions are inhibited at night. Now, the plant still is going to need energy. So at night, the plants behave biochemically like an animal cell. They're going to use their mitochondria. They're going to break down the carbohydrates that they made during the day, just like we will break down our glycogen reserves in our liver at night, because we still need to make sure that we maintain 5 millimolar glucose and we're not eating anything. So we're going to be doing both glycogen breakdown and gluconeogenesis at night to make sure that we maintain our 5 millimolar blood sugar. Plants, they need ATP. They're similarly going to be doing glycolysis. So PFK is going to be active. Does that help?
Yeah, I think I couldn't remember if it was day or night where PFD activated.
Yep. So if you want to just think of it, I'm going to flip back here just for a second. This slide kind of reminds us that the Calvin cycle is not going to be able to operate at night. Okay. So during the day, plants use their chloroplasts. During the night, they're going to use their mitochondria. And it's not like it's totally like that black and white binary sort of situation. But for the most part, they only need to rely on their chloroplasts during the day, but they're going to need their mitochondria at night because they can't make their own ATP using light energy. All right, anybody else have questions on these questions? There's 26 is kind of split there. Plants don't make lactose.
Can you explain what GAP is again?
So GAP is glyceraldehyde 3-phosphate. So if you think about glycolysis, right, that was the end of stage 1 of glycolysis. So by making that 3-carbon piece, we are halfway through, kind of remaking glucose. And in reality, we're past the hard part because we're just sort of, and it's not called gluconeogenesis in plants per se. They just are able to make GAP, which they can use similar enzymes to make it into glucose so that they can then couple it with fructose to make sucrose, couple it with more glucose in alpha 1,4 glycosidic bonds to make starch, or couple it with beta 1,4 glycosidic linkages to make cellulose. So that's where all of this
So that's one of the products of the Calvin cycle, kind
is
of?
GAP.
Okay, yes, yeah.
Yep. So just to kind of flip and highlight where that is. So here's sort of the overview of the Calvin cycle. Is we take carbon dioxide in, we need some money, we need some electrons, and we're going to generate GAP. And the net is that's going to allow us to generate carbohydrates.
Got it. Thank you.
Mm-hmm.
Could you go over 19?
So the reason that mitochondria are able to have a smaller proton gradient, let me just show you a slide first, and then it's actually this previous slide. So do you remember when we were learning about ATP synthase and we were talking about kind of the magnitude of the proton gradient? We saw this equation in mitochondrial electron transport. And again, not an equation you need to know, but what you need to remember is that there's 2 parts that are contributing to the thermodynamic driving force in mitochondria. It's the difference in concentration of protons. And so that's this concentration gradient. But the fact that we also can build up a charge gradient adds like an extra bonus to that thermodynamic driving force. Okay. And this is not possible in chloroplasts because that inner membrane is permeable to chloride and magnesium ions. And so as we pump protons out, let me show you because I just remember seeing it really quickly here. This is on the electron transport homework, but it showed, it's kind of diagramming the parts of a mitochondrion, and it shows as we pump protons out on the inner membrane space, we're accumulating a positive charge out here. Now, with chloroplasts, we're going to sort of negate that because we're going to move magnesium ions in. So we're not going to be able to build up a proton gradient. So we need to have a greater pH gradient to have the same ATP synthesis potential. Okay, so let's look at the answers that we had here. So mitochondrial are able to have a smaller proton gradient than chloroplasts because they are able to generate a charge gradient. Okay, so that's why they're able to do it. They can generate a charge gradient. They don't use a different ATP synthase. It's like, it's exactly the same. We just literally call it a chloroplast ATP synthase. Light discharging a proton gradient really doesn't have anything to do with mitochondria. And it's not, electrons don't begin at a higher level. That's not necessarily the reason that they can have a smaller proton gradient. Is that the one that you picked?
Yeah, that's the one that I picked because I just remembered that in the electron transport chain, NAD or NADH, I remember being like specific that's a high energy molecule. So I figured since the electrons came from there and
So
proton,
remember, yes, they start out high energy, but it's not necessarily that they're going to be higher energy than the electrons that we have in chloroplast electron transport. They just don't start out there. We're going to use light to make those electrons high energy in chloroplasts. But I can see where that could be confusing with that problem, because you're thinking that they do begin at a higher energy level. All right, we went through this GAP one. Here's the last 3 questions. Anybody need me to go through or explain any one of those? Everybody good? All right. So we've got probably about half of our time left. What can I do that would be helpful? Is it going through homework questions? Do you want me to flip through slides and see if there's anything that you're like, oh yeah, can you talk to me about this? What do you want me to do?
there is a specific question I had, but if
Yeah.
anybody else had something else to say, they can say it.
Well, why don't you start, Skylar, and then we'll see if anybody else has anything.
Can you explain the transporter like shuffle thing? Just because there is a lot that was written on that slide, and I don't think video made. So
You are
just
right. You are right that there wasn't a video. So let's see, this was here. So this guy, this shuttle, this guy, or was it this one?
well, aren't those two kind of like
They're connected. Yep.
okay?
So
I mean, it was yeah, the shuttle one was a little bit confusing.
okay, so the idea is if we're thinking about the players that we have in electron transport in mitochondria, right? We've got electrons coming from NADH and FADH2. And then we've got the fact that we're going to be making ATP all the way on the inside of the matrix. So I'm going to flip here just for a picture to remind us, right? ATP gets synthesized all the way in the very inside of the mitochondria, but that does nobody any good if it's all the way inside there. So ATP needs to get out. In order to do that ATP synthesis, we're going to have to get ADP in. So we're going to have to kind of get ATP out after we make it, ADP in, because it's a building block for making ATP. NADH must deliver its electrons on the matrix side. So if we have any NADH that's made out in the cytosol, we're going to need to find a way to get its equivalents in. Remember, we don't have transporters for NADH. And then we also need to get inorganic phosphate in. And even though I talk about FADH2, we really don't have a transporter that we're going to use for that, because remember, FADH2, we're going to see this in the current learning unit that we're in. When we talk about FADH2 being made in lipid metabolism, remember, when we're breaking down lipids, we call that beta oxidation, and it happens in the mitochondria. Part of what we're going to use is complex 2 in the electron transport chain. That's going to be doing that chemistry to convert an alkane to an alkene. So we're going to see this guy again. Okay, but let me go back to where we were here. Okay, let's start here. So again, these are the different players that we need to think about getting them to their right location. So you're good with that, right? So, and I know this gets a little bit confusing because it's like, oh, what are all these 1s and 2s and all of that? Keep in mind that not every NADH is made in a place where it's not needed. When we talk about pyruvate dehydrogenase chemistry, the citric acid cycle, here's beta oxidation, even though we hadn't learned about the details of it yet. All of those are actually made in the matrix of the mitochondria, so they're where they need to be. It's really only NADH that's made in the cytosol, which is only from what we've learned from glycolysis. So let's take this guy first. NADH, we do not have a transporter for NADH, but what we can do, let me zoom in here because it's kind of small here, is we can take the electrons from NADH and pack them onto oxaloacetate. So we can oxidize NADH, it gives up its electrons, oxaloacetate becomes malate, it becomes reduced. So we can think about the drug really being the electrons. And so what we're doing is we're hiding our electrons by converting oxaloacetate to malate. We know that we've got a malate transporter. So we get malate to go across, it's delivering those electrons, and then we reverse this reaction, take malate, make it into oxaloacetate, NAD plus becomes NADH, and now the net is that we've got NADH inside the mitochondria and in the matrix, and it's where it needs to be. But that only needs to happen with NADH that's derived in the cytosol. With me? Here's our revolving door piece. So this is like a big, and I want you to think about it like a revolving door. It operates by facilitated diffusion. So ATP is going to, I should say ADP needs to go from the cytosol into the matrix because that's where it's going to be used as a building block. And then once we make ATP, it needs to go out. Now, the good news is that these are likely to move in the direction that they're needed, because if we're going to be doing ATP synthesis, we're going to be making a lot of ATP out here, and it's going to easily flow out because we don't have it out in the cytosol, which is where we need it. And then similarly, so this is just a revolving door that's going to sort of work in a facilitated diffusion way. Now, there's also this charge component. Don't worry about that because that can get confusing, but just remember that if we're putting ATP out, that actually is negatively charged. It's going to be -3 charge going out, and then ADP is -2 charge coming in. So the net is actually moving -1 out. And that's actually facilitated as well by the fact that it's positively charged out here. So that helps to move that. Moving positive charges out is going to be harder because we're already moving protons out. So we're kind of moving it in a direction that makes sense. Okay. Last piece is thinking about moving inorganic phosphate in. Okay. And this one, we have to piggyback it, because we're actually moving minus charges in here, okay? But what we can do is we can use a little bit of that proton gradient to kind of have a symport here. Remember, symport is a proton or a transporter that's going to move two things in the same direction. But in this case, we're going to use a little bit of the proton gradient's energy to move this downhill so that we can move this guy uphill. Okay, so just call that piggybacking. Another way to think about a symport is that we're piggybacking because we're using this guy's energy to move this one against its concentration gradient. So that's those three things. So hopefully that sort of helped clear that up.
Yeah, it did. Thank you.
Okay. All right. So that was Skylar's question. Anybody else have anything specific you'd like me to sort of clarify, go over, look at in the notes?
Will you talk about the FBPase and the fates of FD?
Oh, a ferredoxin? Yep. So let's go over here. And again, I know what slide I'm trying to get to. Okay, so I want to talk about it on this slide, right? Actually, let me go here because I sort of show it here as well. Okay, so here is ferredoxin, and sometimes, and I'll apologize if it sometimes looks like I write FES when I'm talking about ferredoxin, that's because there's an iron sulfur center. That's kind of the electron carrying part of ferredoxin. So there may be a couple times in different notes or homework that maybe it says FES, which is sort of the active part of ferredoxin. But ferredoxin, once it gets its electrons, so we can see here in blue arrows shows the flow of electrons. Once ferredoxin gets electrons, there's 3 places it can take its electrons. If we have what we call non-cyclic electron transport, it just takes them on to the next carrier, which is this ferredoxin NADP plus reductase enzyme, which we also call FNR for FNR. This is sort of where the last kind of stop is. So I guess, Lauren, if we wanted to think about a homolog for you, because you were complex 4, and that's where the final electron acceptor happened in mitochondrial electron transport, that's kind of an equivalent here, but this is not a complex that sits in the membrane here. But so what happens here is we're going to deliver those electrons to NADP+. We're going to grab some protons from the stroma. We're going to contribute to the proton gradient. We make NADPH, and then we're done. So that's the non-cyclic electron transport. Ferredoxin delivers its electrons to FNR. Okay, so that's the first one that we have here. The other one is it's like, hey, how about we sneak back in and we do this again? This is the cyclic pathway. Not every photosynthetic organism has the ability to do this, but for those that can, ferredoxin is a mobile carrier, and it can say, hey, I'm going to dissociate here, and I'm going to go back, and I'm going to deliver my electrons back to Q, because that's pretty cool, because when we have Q interacting with, in this case, it's called cytochrome B6F, that's where the Q cycle happens. I can translocate more protons, and I can contribute more to my proton gradient, so I can increase my ATP synthesis. With me? So those are the two places where we can see electrons sort of going, cyclic versus non-cyclic electron transport. Now, the other thing is that if ferredoxin is sitting here with electrons, and keep in mind, this is not like one ferredoxin doing all of these things. It's a pool of different ferredoxins. Some of them may be doing non-cyclic, some of them may be doing this redox cascade piece. So if we think about this, if there's a ton of electron transport going on, there's a good chance that most ferredoxins are going to be in the reduced state, meaning they have electrons. With me? Okay, so here's where we can come to this redox cascade piece, which has to do with how we think about regulating sort of the Calvin cycle. So we just talked about going to FNR, that's the non-cyclic pathway, taking them to Q, that's the cyclic pathway, riding the ride again so that we can increase our proton gradient and increase ATP synthesis. The third option is some of ferredoxin in its reduced state, so with electrons, can take them on to a redox cascade. So I'm going to zoom in over here. So here we can see photosystem 1, once the electrons leave photosystem 1 and photosystem 1 goes back to an oxidized state, means it doesn't have electrons, photosystem 1 delivers its electrons to ferredoxin. So again, ferredoxin is going to be in its reduced state. So this is sort of the starting point, ferredoxin having electrons. And we call this sort of like this electron hot potato because it's really passing the electrons just to different species. And I said, don't worry about knowing the names that we have here. But you should be able to think about, if I give electrons to a species that has 2 cysteine residues in a disulfide state, that's an oxidized state. So if I deliver electrons here, I can reduce that and I can get to a thiol, for 2 free thiols on my cysteine residues. Does that make sense, Lauren? So I'm toggling back and forth between an oxidized state of my cysteine residues when they're disulfide linked and a reduced state when they are in their free thiol form. So we didn't talk about the names of these enzymes, and you don't need to know them, but there's a couple of places where we're going to have electrons go to. So ferredoxin tags and gives its electrons to this protein called ferredoxin thioredoxin reductase. This guy then gives its electrons to something called thioredoxin, and thioredoxin is going to go give its electrons to either FBPase or SBPase. Those are 2 important enzymes in the Calvin cycle. So if you want to know the important enzymes that we have in the Calvin cycle for sort of regulating, remember we looked at this, it's Rubisco, and it's SBPase and SBPase. So here's how we control these enzymes. Rubisco is controlled by pH, by magnesium, and by a transition state analog. So when pH is high and magnesium is high, we're going to be activating Rubisco. If it's dark out, pH is low, magnesium is low, and then we're going to be synthesizing a transition state analog that's also going to make sure that Rubisco is quiet. Now, it's easy to keep FBPase and SBPase quiet at night because ferredoxin is not going to have electrons if electron transport isn't happening. Electron transport won't be happening at night because there's no way to excite the electrons to have them go through electron transport. So in this case, if this redox cascade is quiet, then neither FBPase or SBPase will be in their reduced state. If they're not in the reduced state, they're not active. Okay, and the reason that we sort of summarize, it was a great way to review because this is something we're going to see again, but this phosphorylation cascade is a way to connect an external signal with a downstream response. That's what this redox cascade is doing. The external signal is it's light out, and if it's light out, you want to make sure that the Calvin cycle is doing its chemistry. And by having a redox cascade, you ultimately have this last guy in this cascade be in a reduced state, which means it's active.
Thank you.
That help?
Mm-hmm.
All right. What else can I help explain a little bit more? Let me see if I can get these all on one page here. Just going to flip through and see if there's anything. So make sure, like, know these two reactions that are sort of the net reactions that we have for the light reactions and then the Calvin cycle, also called the dark reactions, also called the light independent reactions, right? But understand how these two equations connect with one another. So these processes are coupled, okay? We went through that guy, how light is harvested, kind of comparative electron transport. So again, for both of these, I would know these pathways for electrons, like where do electrons start? Ultimately, for chloroplasts, they start with water. Water's oxidized at the oxygen evolving center. It's ultimately going to replace an electron that, was ejected from chlorophyll, but the net is water is going to provide electrons that get excited in chlorophylls. They're going to go through this electron transport chain. They're ultimately going to get excited a second time at photosystem 1. Then they're going to go to ferredoxin, FNR, NADP+, final electron acceptor to generate NADPH. So know sort of what we have in black here. Those are our electron sort of pathways. All right, because that was, if I'm coming back over here, there's two questions for electron flow. One's for mitochondria, one's for chloroplasts. So be able to diagram and show me that you know the pathway for electrons that are flowing. And then the important equations that happen in chloroplasts and mitochondria. So again, these are the important equations in chloroplasts, right? So the light reactions and the dark reactions. And then this is the important equation in mitochondria. Okay, so knowing those are going to give you what you need essentially for all of these questions. Other questions? Let me just flip through and see if there's anything else to highlight. It's funny, I ran into Dr. DeLance. We had the Pennsylvania Academy of Sciences had their meeting. And he said, it was funny because this was Friday night, we had a board meeting and then there was like a reception event. And he said to me, he's like, totally strange question. He's like, so in my class, we're talking about the enzyme Rubisco. He's like, are you familiar with Rubisco? And it was so funny, right? Because we had just talked about this on Friday. And he's like, I'm trying to understand with RUBP. We started talking about things. And then I started talking to him about this whole concept. Oh, he was trying to understand and explain why in the heck would Rubisco want to have oxygen also as a potential substrate. And he's like, it just doesn't make sense. So I just sort of talk about it as it being like this quirky, like part of evolution that it could take one or the other. And I was like, no, it's all about making sure that Rubisco has something to do. Because if Rubisco gets inhibited and you pinch the pathway with Rubisco and electron transport is still going on, you're going to have reactive intermediates that are going to kill the chloroplasts. And he's like, oh my gosh. He's like, why don't any biochemistry or why don't any plant textbooks talk about this? And I don't know, maybe they don't understand their biochemistry well enough. But he's like, it totally makes sense. He's like, all I've been doing is telling students that it's like, it's just this weird quirk in evolution. And I'm like, no, it totally has a purpose. Usually there's not weird quirks in evolution. Something happens for a reason, right? And that's why photorespiration happens, is to make sure that Rubisco has something to do. Because if Rubisco doesn't have something to do and it gets kind of gummed up, that's going to kill chloroplasts and that's going to kill the plant. He was like, whoa, that is pretty cool. So I should find out, there's one video, and I think it's this video that there's like a ton of people that have watched. He's like, can I show? Because I showed him the YouTube video and he's like, can I give that to my students to watch? I was like, yeah. So there's like one video I think that has way more views than any of the other ones, which I know are you guys kind of watching them periodically. Anything else? I don't want to stay on just to stay on for an hour, but. All right. So the plan is that we will start at 8:30. I know, Adam, you said you can't be there till 9. And if you feel like you want time past that 50 minutes, you are welcome to stick around until 9:30. Okay. I'll check in with you that day and see if that's kind of what you're feeling, because then I won't have you start your 10 minutes, because your 10 minutes, your 15 minutes has to kind of be at the end of your exam. But we'll plan on that. And then let's plan on 8:30 as well on Thursday, because I want to sort of have that be a standing time where we get together, we sort of talk about research plans, and we'll kind of set the schedule for what we want to do over the next week, which for this next upcoming week is going to be a little bit wonky, only because I'm going to be gone in Seattle from Friday through, I won't be back till Wednesday morning. So, but yeah, I'll have a video for you guys just to watch as sort of a flipped lecture for the topic that we would talk about normally on Monday. All right. I don't have anything else. I'll stick around if anybody has any questions, but we'll kind of, we'll call it a night and give you guys an opportunity to get some studying done. All right. See you guys.
Thank you.
All right. You're welcome. Bye.